Hi everyone, this is lecture 10 and the topic is functional adaptation. An adaptation is a biological mechanism whereby organisms adjust to new environments or to changes in their current environment. It could have several interpretations or meanings. It could be a process that will fit the organisms to their environment. It could be a state that has been reached by a population during the process, or it could be a phenotypic or adaptive trait with a functional role in each individual organism that is maintained and has evolved through natural selection. But what is functional adaptation? A functional adaptation is a structure or behavior that has arisen sometime in the evolutionary history of a species to aid in their or in their predecessors' survival. In functional adaptation, structures that increase the likelihood of survival are strongly favored. These structures appear in great numbers from one generation to the next and becomes a fixed trait, such that all members of their population would have this trait. Convergent evolution is when these similar traits are fixed in different lineages. In this figure from Lee and Coop, 2019, it shows the allele frequency trajectories, there in red, at a single locus for two or more populations. So you will see that in A, the beneficial allele was standing in the ancestor of three populations where it was fixed in populations 1 and 3 but lost in population 2. Onsets of selection marked by the blue triangles. In B, the beneficial allele was standing in the ancestral population of our present-day populations. Its frequency increased when the populations split. Then it continued to increase in both daughter populations to fixation. In C, the beneficial allele arose in a single present-day population and spread via introgression into the other population. In establishing a case of convergent adaptation, two main conditions must be satisfied. First, the character must be adaptive, so it has to enhance the fitness of the organisms that bear it relative to the ancestral state, and that directional selection repeatedly drove the convergent state. Second, the adaptive changes in the different populations must be independent from each other. In quantifying convergent adaptation, this can be done by assessing the degree to which selection has independently driven phenotypic and allele frequency change. And lastly, population genetics can be used to identify convergent adaptation. In divergent evolution, the accumulation of differences are seen between closely related populations within a species, leading to speciation. We've discussed this before in speciation. So this would be exhibited when two populations become separated by a geographic barrier, such as in allopatric or peripatric speciation, and we have discussed this before. And they experience different selective pressures that drive adaptations to their new environment. Adaptive radiation is a process in which organisms diversify rapidly from an ancestral species into a multitude of new forms. So this produces a wide variety of types adapted to specialized modes of life. And of course, this diversification results into novel biological communities. In terms of functional adaptations in amphibians, so there have been studies that show a tight correlation between the phenotypes and the habitat types. So for example, there is a suggestion that there is a correlation between body size and ecology in the study of the Philippine fanged frogs occupying similar or more general ecological niches. So for example, the large-bodied Philippine fanged frogs Limnonectes acanthi, Limnonectes furneri, Limnonectes macrocephalus, and Limnonectes magnus and visayanus are found in medium to large rivers. And then the medium to small-bodied species, for example, like Limnonectes latensis, Limnonectes parvus, and Limnonectes palavanensis are found in swamps, sips, and wide, slow-moving streams. In the study of Blackburn et al. In 2013, they found a diverse clade of frogs, the Calula genus of Microhylidae in the Philippine island archipelago, and that the adaptive radiation were found into 3 primary habitat specialists or ecotypes. So in terms of the habitat, there were the obligate forest species and then species common in open or disturbed habitats. For microhabitat preference, ground species, species that perch when active, species exclusively found in tree holes were the different forms that were found, and that the morphometric data were used to have a general shape for each species. So this maximum likelihood phylogram, of course, this shows the phylogenetic relationships of these frog species. And this was estimated from mitochondrial DNA sequences, the 12S and 16S ribosomal RNA genes, depict the phylogenetic relationships of Calula from order Anura family Microhylidae. So the gray box indicates the endemic Philippine radiation. And you see there the different shapes, the different forms. And that shows 3 primary ecotypes. One is the arboreal tree hole frogs. The second ecotype is the scansorial shrub frogs. And the third are the ground frogs. And the loading on the traits were related to climbing, which were indicative or indicated through the length and width of the digits and the digit tips. It was also related to digging from the lengths of the metatarsal tubercles. So this suggests that morphological trade-offs involve in vertically partitioning habitats during ecotype evolution. In this paper of Kyriazis et al. On the dynamics of genetic and morphological diversification in an incipient intra-island radiation of Philippine rodents of the family Muridae, genus Bulimus. For rodents like Bulimus, their external features are not that distinct, and traits that are usually analyzed are skull and dental measurements. For this study, they used 16 craniodental features for Bulimus luzonicus from various populations. And then this maximum likelihood phylogeny analyzed 192 Bulimus specimens based on 1,143 base pairs from 23 study areas, mostly from Luzon, but it also includes those populations from Mindanao. And their study showed that Bulimus in Luzon Island appears to be undergoing speciation. So maybe we can expect in the future that there would be several populations that will be elevated or described as new species of Bulimus, distinct from Bulimus luzonicus. For the polytypic species, and this is a, maybe you are familiar already with this, the Philippine hanging parrot. It's found throughout the Philippines, Loriculus philippensis, and there are different subspecies that are unique to single island or group of islands. And so there are several subspecies based on their size, the color of the bill and the feet, and in terms also of the plumage patterns. And just to get back at what we have already studied, so what is usually used is the phylogeny-based species delimitation. And this is the usual basis for taxonomic revisions. And the reasons could be due to increased sampling or due to robust molecular data sets. So the kind of taxonomic revisions seen are subspecies that are elevated to the species. There could be splitting. There could be a redescription, or there could also be renaming. Now for your learning activity, which is also related to the other topic on taxonomic revisions, kindly read this article, which was published in 2006. So this is by Tello et al. The article is entitled "A New Species of Hanging Parrot from Class Aves, Family Psittacidae of the genus Loriculus." So from Camiguin Island, Philippines. And explain the rationale and the method of taxonomic revision in this case. Well, technically, this is not a new species anymore because 15 years have already passed since it was described. However, when I search for Loriculus camiginensis in the IUCN Red List of Threatened Species, there is no search result. And a search in Avibase, the World Bird Database, so you see there Loriculus, and then enclosed in parentheses Philippensis, and then Camiginensis. Okay, so why do you think is this the case? Were there attempts to also elevate other subspecies of Loriculus philippensis? And what may be the reasons for the attempts or the lack thereof?